use a laser pointer because if you use a laser pointer, it will online again. All right, Alexa says we should get started.
Okay. I'm going to need that. It's too dim without the light.
We'll keep it on until the middle.
Should we go ahead and get?
Yeah, go ahead, get started.
Okay, everyone, we're going to get started. So welcome to our final BME seminar for 2021, hosted by the Department of Biomedical Engineering at Columbia. Wow, I didn't know that was applause already, but okay. So my name is Aaron Kyle. I'm a senior lecturer in biomedical engineering design here at Columbia, and it's my distinct pleasure to welcome today's speaker. We are going to continue offering this lecture in a hybrid format with both virtual and a live audience. Our speaker will present for approximately 40 to 45 minutes, and we will leave 15 minutes at the end for Q&A. For our virtual audience throughout the presentation, please use the Q&A feature at the bottom of your screen, not the chat. So again, use the Q&A, not the chat, to submit your questions. We will try to answer as many questions as possible during the Q&A at the end. For our live audience, you will have the chance to ask your questions during the Q&A session as well. So without further ado, it's my distinct pleasure to introduce Professor Maribel Vazquez. We are excited to welcome her here at Columbia. And it's great to have somebody that's not only a distinguished faculty, but a longtime friend come and visit us here at Columbia. So Dr. Vazquez joined the faculty of the Department of Biomedical Engineering at Rutgers University in 2019. She was previously up the street at CCNY. She was a professor and co-founder of the Department of Biomedical Engineering at the City College of New York. She began her career in industry working for Intel Corporation and then completed a Doctor of Science in Mechanical Engineering at the Massachusetts Institute of Technology. It's a small college, nobody's ever heard of them, but all right. Her research develops microfluidic devices to examine cell migration and differentiation in the nervous system. Recent projects from the laboratory have begun to prototype hybrid microfluidic ex vivo approaches to promote the integration of transplanted retinal stem cells. Professor Vazquez is also a class of 2020 inductee of the American Institute of Medical and Biological Engineering, my fellow class of 2020 inductee. Please join me in welcoming Professor Maribel Vazquez.
Okay. And you can just make it small, sorry. Yeah.
Okay. You can just use the mouse to point.
Go ahead. Okay. Good morning, everybody. Can you hear me? Is the mic working? Yeah. Yes, awesome. I'm so excited to be here. Thank you so much for the invitation, for the very nice introduction. I am Maribel Vazquez. Today we will be talking about some projects in my lab that revolve around microfluidic modeling of retinal stem cell transplantation. So a quick overview of the talk. I'll give you an introduction to me and to my lab. Talk about the, sorry, let me take this off. Sorry. I can breathe now. Talk about the human visual system and then adult vision loss in the United States in particular. Then switch to a more technical part of the talk to discuss roles of neurons and Mueller glia in retinal structure, function, and repair. And then I'll share with you 4 projects, BME projects in retinal engineering that I've highlighted here. Fear not, I am aware that it is a 40-minute seminar, so it's highlights of these 4 projects. I'm not going to go through 4 individual papers today. And then we'll talk future directions and discussion. So first, some fun facts. This is my electronic business card with all my information on it. Right? Nobody has paper business cards anymore, especially in the years of COVID. No one is accepting a piece of paper that you're taking from your pocket and giving to them. So this is my e-card, has all my information. Probably the most important here is my lab website, vazlab.org, V-A-Z-L-A-B. It's easier to say than my actual email, which has my full name in it. And in a true to form, in this millennium, everything from the time I was born up until this very moment professionally is on that website. So all my research, talks, educational profile, teaching profile, all of that's on there. So take a look. My email, my phone. I point to you here my LinkedIn address, which is the only social media that I have, which my students say LinkedIn is just Facebook for professors. So here's my latest Facebook post. We're hiring at Rutgers for 2 faculty positions next year and also some postdocs. So take a look and come talk to me afterwards if you're interested. This is us at the BMES last year, a hybrid event in Florida, which was great. So some fun facts. Today is actually my second in-person seminar since February 2020. So I'm really happy to be here. I'm happy to be in the hybrid format for those of you who are not at Columbia campus today. I'm happy to see so many people in the room today. Thank you for joining me. And I've been at Rutgers a little bit less than 3 years, and I really like it, but it's interesting because I've been in New Jersey about 1 year and 2 months and working from my New York City living room for 1 year and 9 months. So it's been kind of an interesting experience. But you know, it's working well. As you heard, I am a co-founder of the BME department at City College, just a mile down the road, and I know a lot of the faculty here from way years back. I look forward to talking to more people in the afternoon. And I have a little bit of a different career path, which I like to highlight here, particularly for the students. So I went from undergraduate to industry, from industry to a doctorate program, directly from a doctorate program to a teaching university, and then from a teaching university to a research university. And I show this here to show you that I pretty much did exactly the opposite of what every professor says you should do if you want to be a professor. I did things completely out of order, but the advice I'll give you is that you have to do things with purpose in your career. And I chose to do these things. It wasn't happenstance. And I'm very happy with how my career has progressed. And keep that in mind as you're progressing in your own professional career, that you make your own path and you'll make it work, irrespective of different advice you get from different people, or how different paths of different people. I have started doing a little bit administration again now at Rutgers. I am the STEM faculty director for DICE, which stands for Diversity, Inclusion, and Community Engagement. Particularly in community engagement, I focus on 3 things. So one is education and undergraduate programs, and not just senior design and lab courses, but also REU programs and the co-PI for our REU program in the department. Outreach, which is huge because the USA really needs more STEM majors. And we've been focusing on non-traditional students through K through 12 programs, through older students, through parents, veterans, and everyone in between. We want to get as many people interested in STEM as possible. And I highlight here in red, one of the big foci that I've had in my lab the last 10 years has been community engagement through health disparities, examining our BME contributions in the wider lens of human health and community in our society. And so that's where I'll start, is what are health disparities? So health disparities defined by the NIH are differences in incident burden and treatment of illness based on non-medical factors. So these are things like socioeconomic status, educational attainment, employment. And it's been very well studied by social science and community public health faculty, but not so much by STEM professionals. Now, there are very large health disparities in the US in every system in our body, unfortunately, but particularly in US vision loss and worldwide. And it's actually a global health challenge from the WHO since 2017. I show you here 3 very big areas of health disparities by age, gender, and geography. So for example, those who are 65 and over have a 27 times more likely to lose vision than those who are 25 and under. If you're a woman, you're 6 times more likely to lose vision than a man. And depending on where you live, this even gets even more interesting. So looking here at the map of the US separated by time zones, the states highlighted in black represent states that have 7 times the national average of adult vision loss. And it's kind of strange because when you first see that, I initially thought it would be states that have a lot of radiation. So California, Arizona, not really. The Northeast is doing pretty bad. We're over here. The only place that's doing worse is the Gulf, save for Georgia. They somehow dodged the bullet, we don't know. And so trying to understand why that is, that we have these geographic disparities as well. So I've introduced this in my lab and in my education and outreach. The goal is to introduce health disparities in STEM training and in BME research. So I've done this by creating different courses and capstone projects at the interface of STEM and health disparities, training students to select cell and animal sources that are actually representative of the disease that we're looking at, whether it's a genetic model or a clinical trial, and identifying the environmental conditions and control conditions. Is it a genetic disease? Is it an epigenetic disease? Also investigating imaging. So imaging based on anatomical structure, how we see the eye, how your actual physiology for your own body is different from the average and how that affects diagnosis. And the biggest is increasing awareness of health disparities among STEM professionals and also developing ethics in ethics in development of therapies we're developing today for the future and how we implement those therapies. And if you're interested, I point you to ebix.net, Emerging Behaviors of Integrated Cellular Systems. It is an NSF center that I've been part of. This is the Swan Song year 11. So this is the last year. The PI is Roger Kam from MIT. And on this website, we have many different vignettes that you can look at for ethics, which I think is appropriate for K through 12 all the way to faculty and beyond. So some very interesting questions of how would we develop this particular therapy and who are we trying to help and who actually gets hurt by this? And to think about those questions before we start as engineers actually making the technology to implement these therapies. So I point you to that website. But of course, we can't really talk about vision loss without talking about vision. You know, what is vision? So I'm just going to move this menu. It's totally in front of me. Here we go.
You can,
If you click on more, you
Yeah,
can click
here.
on.
I can,
Thank you.
sure.
2 years and we're still not
Yeah, I know.
professionals in that.
Just practice this. So here we go. Ta-da.
Oh, thank you so much. I'm going to use that next time I give a talk because I didn't know I could do that. Okay, so what is vision? So vision is the conversion of electromagnetic radiation or photon particles, AKA light, depending how you want to consider light, into electrical signals that form an image in the brain. The visual system is comprised of the eye, the optic nerve, and connecting pathways to the visual cortex in the brain, things like the optic chiasm, the optic tract. There's 25 million adults with impaired vision in the United States, and there's a 50% increase expected by 2050, which is like tomorrow. This is an incredible increase, according to the National Eye Institute of NIH. We're talking about visual impairments and not squinting, right? So this is not glasses. This is actually losing your vision, and it's very complex and can occur in different parts of the eye. We have different components of the eye. So let's do a quick primer on our eye, shall we? Okay. There we go. So in the front of our eye, we have the cornea. And the cornea, in the cornea here, you see the incident light shown in blue is collected and refracted into the eye by the cornea. Light enters through the pupil, the black of the eye, is focused by the lens onto the focal plane in the back of the eye, which is the retina, which is right here, this mustard colored thing here. So the retina is the focal plane of where the light is focused into the eye. The retina transduces light into chemical signals to the brain for vision. So it is our photonic organ. Dysfunction in any component in the eye, however, can lead to irreversible vision loss in many different ways. So for example, so you have to, how would that affect what we see? So if I show you this image, and this is not my work, but I'm show you the citation here. So what I see in this image is a woman with very colorful headscarf and colorful blouse. This is what I see. Depending on different types of visual impairments, you can see the same image in different ways. Right? These are 3 very common visual impairments in adults. The first one, A, is glaucoma. So this is damage to the optic nerve through hypertension. So it kind of flattens the back of the retina. So you keep your central vision, but lose your peripheral vision. B is cataract. So this is clouding of the lens by degraded proteins in the lens that then scatter and block light. So it looks like you have just a constant film, which you do, in front of your eye. And then C could be representative of 2 different diseases. One is macular degeneration, which is the thinning of the macula, which is the center of the retina, we'll talk about in one moment, to damage the photoreceptors, which are rods and cones. These are the cells that actually transduce the light. So when these cells die, what we see is pure blackout. It's not blurry or stigmatism, it's just black because light is not being converted. This also could be from diabetic retinopathy, which is when the vasculature in the retina is actually damaged because of the high glucose levels in the blood and drains into the blood. Note for C, you don't necessarily have to have the blackout in the center, and it doesn't have to be a big area. It could be multiple little black punctate spots in your vision throughout the whole field. So there's a growing focus on epigenetics and age-related vision loss because these 3 most common ones worldwide, in the US as well, are closely related to age, 65 and over. So we'll talk about adult retinopathies. Now, age-related retinopathies are rising because of increased longevity in high and middle income countries. Show you here the three common ones we just discussed. What's expected worldwide, millions of adults visually impaired. Look at this number, 275 million worldwide expected from macular degeneration. And right now we're at 185 million. Diabetic retinopathy is less so, and glaucoma, but we're expecting significant increases. Macular degeneration, or sometimes called age-related macular degeneration, called AMD, are among the fastest rising disorders in the United States. And that's what my lab has focused on, is AMD. In the US in particular, though, our obesity rates have produced exponential increases in numbers of patients with diabetes and other metabolic syndromes. This just came out this week. 42% of the US population is obese. I'm not talking overweight, we're talking obese. Body mass index of over 30%. Right? This is an incredible jump in the last decade. And because of that, we see the millions of adults with diabetic retinopathy, how it's going up by 2050. You can see that's why it is a severe challenge that is already on our border of time. So neural cells have become therapeutic targets because of their roles in retinal metabolism. So we're also working on diabetic retinopathy because we're looking at regenerative therapies for the AMD and the diabetes. So here's a cross-section of the human retina shown here. So this is a cross-section in the back. I remind you again that the retina is this entire piece. It's like a moon crescent shape in the back of the eye that connects to the brain through the optic nerve. And this is a cross-section. So if we take the cross-section, it's about 250 microns at its thickest, which is the back, could be 12, 14 at its thinnest in the front. It has over 100 million cells. It is the most cellular structure in our bodies. And the retina, the human retina is arranged in three nuclear layers, the neuroretina, the outer, the inner, and the ganglion here. So this is the ganglion, the inner, and the outer. And two plexiform layers, which are the areas in between where the neurons actually synapse and transmit the signal, the photonic signals. It contains 5 types of neurons and 1 glia cell called Mueller glia. And I tried to color coordinate the names with the pictures here. So the Mueller glia, abbreviated MG, are these blue cells that across the entire thickness of the neuroretina. They ensheathe neurons and vessels, and they protect these cells and also regulate the inner blood barrier, inner retinal blood barrier, outer retinal blood barrier, and regeneration of the natural repair of the retina. The photoreceptors here, rods and cones, are the only cells capable of phototransduction in the retina. And so despite all these other interconnected cells, these are the only ones that are actually capable of phototransduction. Looking here from a rod cell, we're talking about taking in light, being absorbed, activating rhodopsin, transducin, and then the cascade. When these cells die, they cannot self-repair. So then we have the blackout punctate spots. It's like a light bulb burning out. So mature retinal neurons are unable to self-repair, and because of their interconnectivity, they really propagate synaptic dysfunction very fast, leading to vision loss, eventual blindness. A transplantation of retinal stem cells offers promise to restore vision because we can actually replace these damaged cells. So so-called cell replacement therapy transplants stem or dedifferentiated cells or progenitor cells into adult retina to replace these damaged neurons. Now, the eye is an excellent target for transplantation therapy for a few reasons. One, the micrometer dimension is 250 microns, enable study of this process using microliter volumes of reagents and lower numbers of replacement cells. So we don't need 100 million cells. We don't even need 1 million cells because of the scale. Right? If we can get 50,000 cells to actually migrate into the retina, we can actually cure blindness, I think. So also ophthalmic imaging really allows us to evaluate stem cell delivery and integration of the cells that are transplanted. And particularly looking at photoreceptor transplantation, which is what my lab focuses on, is advantageous because the photoreceptors here have only one primary synaptic target, which is the bipolar cells. Every other cell connects with a different type of neuron on each side. Only the photoreceptors have the synaptic connection on one side. And so I show you here a cartoon of transplantation where we take stem cells shown here in blue, put them in a scaffold of biomaterial or a different type of matrix or liquid, put them in the subretinal space, and hope that these cells will then migrate in to replace these degenerated cone and rod cells. Not these two, but somehow figure out how to get to these 3. So if we were to simplify the transplantation process into 4 steps, it would be these. One is survive surgical incision, because these are stem cells and they're very finicky. So they have to survive the the incision, the implantation. 2, they actually have to infiltrate into the retinal host. And so it's not just moving, it's actually integrating through, if you're looking at, think of 100 million cells and you're coming from the top and you have to integrate between these columnar photoreceptors. 3, they have to differentiate and position appropriately because they have to actually, then step 4 is synapse with this cell. So not only does it have to come from here, figure out that it has to go in this direction, it has to change its morphology to then synapse with this and somehow differentiate. Not hard, right? Not at all. Each one is taken, hundreds and hundreds of research labs to get to this point. But I'm confident that we will cure this disease in my lifetime. But what my lab studies particularly is infiltration. So we're looking at infiltration of these transplanted stem cells into adult retinal hosts. So our lab seeks to increase the effectiveness of this cell replacement therapy by modulating integration of these transplanted cells and also looking at the neuroprotective responses of the host cells, particularly the glia, but I won't talk about the glia projects today. So our experiments use microfluidics and explant systems. Spoiler alert, I am a microfluidics gal. We're going to talk about lots of devices to at least show you cool pictures of them. And we apply different controlled stimuli to examine cell extension, migration, communication between neurons and with glia. So the 4 projects that I'd like to talk to you about for the next 25 minutes or so is looking at chemotactic responses of these transplantable cells, their collective behaviors, modulation of their behaviors using external fields, particularly concentration gradients and electric fields, and then end with some agent-based modeling or computational modeling of transplantation, the infiltration of transplanted cells. So we've used Transwell assays or Boyden chambers, microfluidic assays for migration, nanofluidic assays to study extension, and then more recently having this ex vivo assay with a whole enucleated eye. So I'm a microfluidics person, so I have to, of course, say how microfluidics is the best field ever in all of BME and engineering in general. I'm sure you all agree. Right, so microfluidics is a field that manipulates and monitors behavior of fluids within geometrically constrained structures. Those structures have to be less than 1 millimeter on a side. So 999 microns and below, you too can call yourself a microfluidics person. So microfluidic systems enable precise tailoring of analytically defined environments that are extracellular and allow us to measure cell behavior in response to that controlled stimuli. These devices can provide a variety of external stimuli such as flow, diffusion, electric fields, mixtures, any combination. And these devices can be really micro-manufactured now pretty cheaply, pretty quickly over the last 20 years, can be closed, open, surface pattern, 2D, 3D, multiplex. I'm sure we've all seen the devices that look like Ruth Goldberg machines, some of micro devices because they're so complex. But we can also make ones that we just micromold using PDMS and make them very straightforward so that we can share them with things like people like clinicians who don't have to understand the transport inside, just how to use it and how to image it so that we can have collaborations to answer some very fundamental biological questions such as migration. So I'll show you one of our more straightforward systems called the GLL or the GLine system, the GLIAline system, because we developed this to study Mueller glia, which are the biggest cells in the retina. Here's a device here in a simulation. Here's the actual device stained with, filled with a red dye. It is 2 reservoirs here that regulate this microfluidic channel underneath and allows us to create linear or nonlinear gradients of diffusion, of convective diffusion, and of electromagnetics, all the fun stuff that we can apply on the outside of the system. And to give you an example here, the red, in this particular case, we're looking at convective diffusion. Sorry, the green are experimental data, and the black is the analytical solution to the equation. So we're within 1% because of the scale. And it is the big benefit of microfluidic devices that we can model it, solve it analytically, and then compare it to experimentation in such a tight margin. And here are the Mueller glia stained in a dish, just to show you that their morphology stays the same in a regular flask as when they are in our devices. And this cross section is 200 microns, sorry. So doing this gives us a transient development of this gradient, the microenvironment, and allows us to correlate the cell response that we see to the signaling pathway. So just to show you in cartoon format, if we look at the GLL, here it's made of PDMS, this block, and then a glass cover slip. We fill one reservoir with media, and we've done this with many different types of cells. So we've done it with lots of different types of media, from Drosophila cells to rodent cells to human cells. And we fill the opposite reservoir with a chemical stimulant. And this could be any kind of molecule growth factor from 50 Daltons to 300 kilodaltons. And we've done a range of concentrations as well. These two reservoirs are used to regulate this microchannel at the bottom. And this microchannel, we've made it different diameters and different lengths, depending on the system that we use this for, whether it's in the eye or in the spinal cord or in the brain. And our cross sections using this microfabrication of PDMS elastomeric molding between 100 microns and 250 microns. We can make this in the lab very quickly. And this length here, the length of this microchannel, sorry, my LC is not showing up. The length of the microchannel is really limited by the stage of the microscope. So we can see the entire field of view. So we put cells in solution on the media side. We let this concentration, this stimulant transport across, creates a chemical gradient and causes these cells to migrate towards increased concentration gradients. And we have less than 2% error, really less than 1% error in solving this equation. And we see this in real time in the microscope stage. Now we have an incubated stage, so we can keep it running on the microscope for days. This particular device for about 3 or 4 days, and we replenish it and can survive for probably 7 or 8 after replenishment. Or we can actually take this, put it in the incubator, take it back out the next day, look at it, put it in the incubator, so that other students can use the microscope, for example. So we've done a lot of things with these types of devices. From a long time ago, we started looking at different migration modalities at different progenitor cells. So we've looked at up and down regulation of signaling pathways can lead to different migratory responses of neuroprogenitors to the same stimulus. So this here, I show you, this is an older work looking at glial progenitors, and we have 4 different types of glial progenitors. And they are responding to a stimulus, the same exact stimulus of TNF alpha on the right-hand side. And you see for the first cell type, the cells migrate as individual cells toward the right. For the second cell type, they migrate in daisy chains so that they're always in contact with their neighbors as they migrate to the right. For the third cell, they migrate in mass, like wave migration. And in the last one, they actually are chemorepulsed. So these 4 different modalities are seen with the exact same stimulus. Was also particularly more recently, we've noticed that retinal progenitors can migrate individually, collectively, or in groups of varying lineage. Of course, they have to be the most complicated. They can have every single combination there. And so here, show you these are oligodendrocyte progenitor cells. These are retinal progenitor cells. These are medulloblastoma, transformed progenitor cells. And you can see these cells are always collective. These cells are collective and individual, and these cells are always individual. Looking at this, we've been able to stain for reactive cells, looking at GFAP, vimentin, nestin, and how they migrate individually, you see here. And we've also examined a few different cell lines and also some primary cells. And I'll point here that there are very few microdevices that have been applied to study retinal cell response for retinal therapies. It's really been a lot of in vivo work and a lot of differentiation studies for the transplantable cells, which is incredibly important. But I think the complement then is how do we actually transplant these cells and see how they behave in an external environment. So the first project is migration, and this we're looking at Muller glia migration, but looking at reactive Muller glia that actually are the ones that help repair the retina to any type of insult. And we know that these responses, chemotactic responses, have been studied in vivo to a variety of different agents, but we examined their ability to chemotax in vitro to a panel of different growth factors. This is particularly important for later stages of gliosis and could lead into glial scarring, which is if we're trying to transplant cells into the retina, we have to consider how the native tissue will respond to an injury. It'll make a scar. And so even if we get these transplanted neurons to go to the right place, the scar will completely sever that part of the eye from the brain because it is an injury and we have to actually go and insert. So these 2 things go together. So here we use transwell assays, like boarding chambers, 2 wells, concentric wells within one another, cells on the top, growth factor on the bottom, a membrane in between of 10 micron pores or less. The cells migrate from top to bottom through the pores and then stick on the bottom. And then we image the number of cells toward different stimulus. And here in this particular case, we're looking at dosage concentration effects with EGF and to see which concentration has the most chemotactic effect for the Muller glia. And so for this project, we selected EGF for further study. And when we applied this into our micro device here, which is a GLL device, what we see in addition, or enriched from the Boyden chambers, we see a trajectory. So we can see how the cell responds to a gradient, not just to a controlled gradient, not just a Boyden chamber gradient, and we can see the actual trajectory at different time points from start to end. Doing that, we're able to see how many distance of travel per concentration gradient, and importantly, we're able to validate the signaling pathway using different inhibitors. So here we've looked, and I know this is a very busy slide, I show you the citations here. We've used Tarceva and UO128 to actually knock down MEK inhibitors and look at the different pathways, looking at RAS, whether it's PI3K, whether it's AKT, and all of that, and able to show that we can silence genes to link this migration, internal signaling migration to the external chemotaxis. So this was a few years ago. So we asked, can we examine these same things for retinal progenitors and when we do the transplantation? Can we translate this now to the neuron cells? And whereas in the first project looking at Mueller glia, we looked at something that's 250 microns in cross section because that is how those cells are in vivo. When we're looking at transplantable cells, we made a microretina device, right? Where it actually looks like the retina, same shape as the retina, and this bulb here simulates the subretinal space when the surgeon has to pull back the part of the retina to inject the cells. So an older project, we looked at different bioinformatics to identify what are chemoattractants for these progenitor cells, and we looked at their migration inside this system. So here shows a schematic of how these cells are drawn into the system and the concentration gradients they will see in spherical coordinates, r, theta, phi. And what was found was that these cells are incredibly chemo-attracted by SDF1, and they migrated collectively and individually in this device. And importantly, we found that their motility index was about 25%. And that is in line with in vivo studies. When you inject these particular cell type into a rodent, you see that about 25% of them become motile. So we saw that as well. So we think that's something internal with these cells. Now, for the first grant we had for this, we had proposed to do FACS isolation of this modal phenotype and say, hey, if we can just take the 25% that are motile out of many animal samples and inject those, then those would just only be motile cells and they would help transplantation. The problem is that you're looking at 1 million cells, that's a lot of pups. That's a lot of pups to sacrifice to do this test. So we kind of tacked a little bit different. And here I show you to scale what this microretina is for the mouse retina. So this is the human retina, this is the mouse retina, this is a penny, just to show you how hard it is to get these cells in there and how many cells we would need. So we asked a little bit of a different question. Can we leverage collective behaviors that we've seen? Do they migrate? Cells migrate very differently when they're individual than when they're in a cluster. And can we apply external stimuli to get to move these cells more than just the chemotactic gradient before we started sacrificing all the animals? That led to the current grant that we have. So in this one, we're looking at collective behaviors. And when we're looking at stem cells, the whole purpose of stem cells, one of the unique benefits of stem cell therapies is that they recapitulate developmental processes to help adults regenerate tissue. There are extraordinary collective behaviors that are needed to develop cell-cell connectivity in the eye. And the signaling that guides eye development is incredibly well conserved across species. So the fly eye is a seminal model for our understanding of how the retina develops for retinogenesis. And it has typically been, it still is, a purview of geneticists. And I'm sure you've all seen, if you haven't, you can go on Google and just look at SEM images and the horrible things people have done to flies. They have some eyes sticking out, some legs sticking out of their retina, a wing sticking out of many different genetic things, and the animal, the fly will actually develop. And so you can see how the genetics are linked to the actual organism. It is a simple model. It's 800 ommatidia, which is R1 to R8. Each ommatidia is a unit for vision, fully mapped genomics and proteomics. You can see ex vivo imaging, in vivo imaging, and because they're flies, nobody really cares. So you don't even need IOCOC protocols, which I think is kind of sad, but you do this in the lab. So you don't even need to do anything with animal subjects. Now, I do have microscale optic structures, which is quite interesting considering how small that organism is compared to us. And so this, we're looking at the larva. So the third, this is a larval system for this organism. The third instar larva is well known to be the point where the retina develops in the fly eye. And so this is looking at the eye-brain complex in this larva, we take it, we've taken it, we've dissected it, and this is in collaboration with my collaborator who's still CCNY, Venkatesh in biology. And what we found was a couple cool things. One, that the cells intrinsically clustered into groups, we're showing here, and 62% of them were in groups of 6 or more, 27%, sorry, 3 to 5, 27% were 6 or more, and about 11% were individual cells. So that's telling us that there's some native behavior of these cells that causes them to cluster or not cluster to aid their migration during retinogenesis. Another important thing is that the ratio of neuronal and glial cells in this particular larval stage has been constant, about 0.62. And these two things seem like small work, but I tell you, the two master students who had to actually dissect this, there is no work on doing in vitro studies of Drosophila, because it's all geneticists. They're like, why would you do cell culture? Just get some maggots, raise them, and have the fly. No, we actually want to study. So there was nothing in the literature about looking at Drosophila cells in culture. So we had to do a lot of that. So it was very interesting from the BME standpoint, and that it actually correlates with the rodent cells that we've been studying. So we developed another micro device here called the micro-os, because the micro-optic stalk, which is the precursor to the optic nerve. And so it replicates the geometry. Here you see the optic stalk in the Drosophila. And here's our device. These little channels are the same size as these ones. These are short and fat channels, 30 microns, 90 microns. So nice and not 1D, but we can still analyze it analytically and still have very little error. This is what the device looks like. We showed that collective displacements and directionality of these cells really depend on the gradient, meaning we had increased directionality within different ranges of different gradients. So these cells were very specific to different chemicals, which was not a surprise to us, but we really were looking at their collective behavior rather than individual cell chemotaxis, for example. So we started asking, what are the relative contributions of the chemotactic field to this cohesion or cell-cell adhesion that we see in this clustering? And so we're examining the interplay between the growth factor receptor and cadherins, which are responsible for cohesion. And this is a paper that we just published here, looking at Drosophila. We used canoe, which is the E cadherin for Drosophila, and we used N cadherin for mouse retinal progenitor cells. And we see the similar behavior here. Here is the numeric representation of the ICC. We're looking at when we stimulate, what is the NCAD expression or the cohesive strength between the clusters when they're in different fields of different chemotactic agents. So you see this is the control. This is in SDF1, which is our strongest chemotactic agent for mouse, and then the other two. Right? So we see when the cells chemotax the most of these fields, they actually are stronger. They kind of like tuck together as a group and then migrate collectively. So this is very exciting to us because this is something that has not been explored in transplantation, is actually looking at clustered migration, and collective migration is very different mechanistically from single cell migration, and it responds differently to different stimuli. So we asked, how do these cadherins influence the response to chemotactic gradients, and can we leverage this to aid infiltration? This one project. Now, the other thing, if you recall, I said we wanted to, instead of sacrificing all those pups, we looked at collective behavior, and we also wanted to look at external stimuli. So if you're talking about the nervous system, the natural stimuli would be electric fields. So we developed a new device called micro-nano, because it has all three scales in one device, and it applies external chemical fields and electric fields. And you can look at the citation of this is a lot here. The coolest thing about this device is that we can apply the chemical field and the electric field concurrently on top of one another without affecting the chemical gradient. Typically, when you apply an electric field, it makes things hot. So it changes the transport of the molecular diffusion. This device was designed in such a way such that that's not the case. So we can actually look at just chemical fields, just electrical fields, and literally the superposition of the two. And when we did that, we found some very interesting results. These are the cells that migrated to electric fields, to chemical fields, and the combination. So the combination is more than the linear superposition of the two. So there is something going on between the signaling of the chemotactic signals and the electrotactic signaling, which we still don't understand. In the field, we still don't understand what those signals are, and much less how they interrelate. Also here you see they become more directional. So the last piece then is here, what about matrix? So now we have the collective cells, we have an external field, we have them migrating together. What's missing in this whole thing is, well, how about the rest of the eye? Right? There are 100 million cells in the rest of the eye and in the retina and also all the matrix. We can't just discard that. So the project we're doing now, we call it EVES, which is an ex vivo eye facsimile system. So we have a chemical reservoir here, a media reservoir here to kind of mimic that GLL that I showed you before, or that microretina that I showed you before. And then we have this electrode chamber of cathode and anode, where we actually put in a nucleated eye. And we have a transplantable matrix. This was used in clinical studies to transplant these cells. Before we get to the eye, though, we're actually doing a facsimile. So we did a hydrogel bead of the same size of a mouse eye. When you put it together, it looks like this. And then this is what the device looks like when it's running. So the facsimile is alginate. Its dimensions are the same as the eye, with microfabricating 3D print. And we're looking at the infiltration of transplantable retinal progenitors into the spherical biomaterial. In this case, it's the facsimile. And when I put in red the transplantable RPCs, because that means a lot of different things to a lot of different people, we're looking at cells that are positive for a PAX6 marker, which is a retinal progenitor marker, and OTX2, which is a photoreceptor marker. So these are cells that will only become photoreceptors. They will not become other types of retinal neurons. And what we found with this exposure is the same behavior that we saw in just the micro device without the eye. And so it's very promising that this combination of fields is doing something on a signaling level that we need to understand. What we're doing now is actually taking the whole eye out. So we're putting the nucleated eye into this explant system. And to show you here a histology with this mouse that has GFP positive photoreceptor layers here. These are the transplanted cells in the red, and we want to see how these cells actually migrate and infiltrate into this host. And of course, that's what we were doing exactly when we had locked down. And so we're just coming up to speed now. And I think the pups are born either today or tomorrow so that we can actually do this test this week. And I'm studying this in collaboration with Lee Tsai, who's 2 doors down from me at Rutgers. And we're also looking at the effects of TOP2B, which is what he studies, looking at some genetic models, if we can do some manipulation of this topoisomerase to aid migration. Now, the last piece here is I think everything I've said is very complex, and we keep adding more and more layers on top of complex layers. And as much as I love doing experiments and as much as I love making micro devices, at some point we kind of need to funnel our work and kind of give some kind of guidance, right? Some kind of modeling as to where we should go and where's the biggest bang for the buck, or how these things link. So I started a new collaboration with Troy Shimbra, also at Rutgers, and we're looking at agent-based modeling. And ABM defines, it's a different type of computational modeling because it defines individual cells as autonomous agents that are then programmed to follow these rules. And these rules are defined by biological mechanisms or experimental data. Right? So it really is a model to help inform what we and predict what mechanistically is happening. And so if we know that this mechanism is actually the primary one, then it'll change what kind of stimuli we apply to aid this migration. Now, ABM is really well suited for retinal transplantation for a few reasons. The spatial architecture, retinal structure is very well modeled with dimensional accuracy, and that's very important. We're talking about 100 million cells. The spacing between 2 photoreceptors in a healthy retina, center to center, is 2 microns. Right? So we have to get these 10 micron cells when they're transplanted to migrate between a less than 1 micron space and then become a columnar cell. So these morphological changes are very important. Heterogeneity is very well modeled using ABM, incorporates mechanical and chemical stimuli. And the stochasticity, we're looking at adhesion-mediated migration here, how these transplanted cells not only have cohesion with one another, but how they have adhesion with the native photoreceptor cells to actually migrate between these 2 columns. And to give you an example, this is one simulation where this is the transplanted cell, and these are 2 existing photoreceptors, and we're trying to understand how to get this cell in between the photoreceptors and change its morphology without actually staying as a ball and separating the existing host cells. So we want to incorporate these morphology changes, and we want to examine chemotaxis and electrotaxis. So in summary, what we've done is we've developed microfluidic models to give us some data to examine responses of transplantable cells to external stimuli, we've been able to show these highly chemotactic responses and galvanotactic responses. And we've really been the first to look at the collective migration as a way to aid infiltration. What we're currently testing is electrochemical fields on these whole enucleated eyes, modulating gliosis and glial scarring using this external field, and developing some biotunable models for AMD and diabetic retinopathy as our disease models. And with that, I'd like to thank my students here. I have 4 PhD students and 2 BS/MS students who are finishing this year, my funding sources, and I'd like to thank you for your attention.
All right, so thank you so much, Professor Vazquez, for the fantastic talk. Before we get to the Q&A, I just want to remind everyone that our weekly hybrid seminars will continue next semester. We're going to start back up on January 21st. Please follow us on social media to receive the latest updates. You can see our social media information on the slide that Alexis has posted up here. I'm like Professor Vazquez as well, and we have LinkedIn, so follow these.
Our Facebook posts. Right?
Yeah, really, Professor Facebook. Okay, so I see we have a few questions going online, and I'm hoping we've got some questions from the audience. So we're going to do our best to answer as many as possible within the 15 minutes we have here. And if we don't get to you, Professor Vazquez put up her virtual card earlier, so hopefully you can reach out her directly and ask questions. So do we have any questions from the audience?
Hello. Okay. Hi. Thank you so much for your great talk. I was wondering a little bit about how you were able to apply both a chemical and electro gradient without affecting the chemical gradient. Could you go into that a little bit more?
I can. That's a really technical paper that in biosensors, but basically we have different flow ports. We have four different flow ports. Okay, my slides are gone. We have 4 different flow ports into the device, and then in between we have a nano array. So we've played with the different cues on the right and the left and the in and the out, so that we can create a highly nonlinear concentration gradient, so that when we apply the electric field on top, over 12 hours, it doesn't change inside that array. And so that's by using actual convective flows, a balance of 4 pumps.
Do we have any other questions in the room? Okay, so I do see we have some questions coming online. The first one I see here is from Scott Sy, and he's saying, Professor Vazquez, can you briefly explain for the migration project, how can you maintain a constant chemical gradient in the microfluidic channel? If there's no flow, I would have thought that the chemical gradient would continuously change over time, and eventually the gradient would go away after enough time.
Excellent question. That is correct. So it's a quasi steady state gradient, but it remains steady state for 7 days, 7 to 10 days, because the reservoirs act as 2 infinite sources. And we have, if you notice on that slide, we have something called a bridge between the reservoirs that actually gives us a very small amount of convective flow. So that velocity is something like 0.06 microns per second, and it's enough to circulate it so that we have the nonlinear gradient at the bottom, and keeps things circulating for 7 to 10 days before we get to that, before the gradient is depleted.
Very good. We've got another question here from Vaughn Green, and Vaughn is asking, I was wondering if there was a particular reason for using zinc chloride for cross-linking versus calcium chloride.
Hi, Vaughn. Yes, I'm sure you know the answer to that because you helped us do that. So I didn't show it here. So we particularly use zinc because it allowed the diffusion of the agents from the bead to be linear. If we use just the calcium, it was very highly nonlinear and very rapid. So we wouldn't be able to do tests over days.
Actually, Erin, I have a question, so maybe we'll pop into the. So when you have that artificial sort of a orb eye that you're now starting to transplant into the mice, is the idea that this will eventually be an artificial eye, or is this more for just experimental purposes?
This is for experimental, and I was just telling this before. So we're taking the eye out and putting it in the device to test the external field component. But then the goal is to apply the electric field by putting electrodes in vivo into the animal. And then you can actually inject a chemical, which ophthalmologists do anyway, so that you can create the chemical gradient naturally and then have the electrode, one in the vitreous and one in the back of the retina.
But with the natural eye of the mouse, not with,
In vivo.
yeah, okay,
in
yeah.
vivo. The goal of this is to help adults who have a dysfunction in the retina. So that it has to be in vivo. That's the next grant, if not the next one after that.
Makes sense. Okay, we've got a couple more questions. We got some time. One from Fatima Kasim, and they are asking, I'm wondering if your microfluidic method of infiltrating the retina can be affected by endogenous factors within the retina. Endogenous factors can also be present during different concentrations and different disease models. Additionally, how can you specifically target the movement of the stem cells without influencing the movement of other surrounding cells in the retina?
Excellent questions. Those are, there are two parts. First, the endogenous environment. Yes, of course, what I showed you was just one primary chemotactic factor, but we have the ability to mix different concentrations of different growth factors that are in the damaged retina. And that I mentioned to you, we have a bioinformatics study that highlights the 4 primary ones that are in injured eye, particularly for AMD, for rodents in this study. So we can mix those to mimic the disease condition. And I just picked STF1 because it was the highest chemotactic gradient for proof of concept, but we can definitely take into account more of the endogenous chemicals. The second one is how we can measure the stem cell migration without impacting the other cells. Excellent question. That's why we had the ABM modeling to help us do that. However, the photoreceptors are kind of fixed in place, so they are not going to be migrating. They would not be pushed out of the way more than just half a micron, right? There is some movement, but it's very small, and they'll stay within that columnar structure in the host. That maintains the integrity of the retinal tissue.
Okay, and I think we'll go for one more here. Giannis, I'm not going to butcher your last name here. So this one is from Giannis, and it's saying, is it possible to put a link to the biosensors paper? Oh, well, we can go over that later. Let's actually go down to Chloe Dujardin, and we'll say, a question about the implantation of the scaffold. If you implant the scaffold in the subretinal space, would the cells need to cross the retinal pigment, I assume pigmentosa epithelium,
RPE,
to reach their location, and how?
Excellent question. So when you do the transplantation, you can transplant them on the opposite side of the RPE or on the inside of the RPE. They've been done both ways. My collaborator does it on the inside of the RPE because from the surgical standpoint, it's easier to pull that part of the retina out. That is the non-neural retina, and the gauge of the syringe fits right inside. So in that schematic that I showed, it's actually inside the RPE, so it doesn't have to cross that columnar network of cells, which makes things more difficult. Incidentally, that RPE is also generally degraded when you have AMD, because that's part of the reason why the photoreceptors die. So they should be able to cross easily, but I don't want to deal with them, so we're putting it on the inside.
Okay.
To the other question that we said deal with it later. So if you go to my website, vazlab.org, on there it has publications and they have links to every single paper on there.
Perfect.
So and they're all PubMed, so you can go get them from PubMed. And yeah.
Okay. Are we good with questions in the room? Has everybody asked what they want to ask?
I
Oh,
think
we've got
we
one more up top, and I think we've got about 5 minutes left, so maybe we'll go for it.
A lot of great questions from chat. Usually it's the opposite.
Hi, Dr. Vazquez. Thanks so much for this awesome talk. I was wondering, I've been doing some reading on engraftment of lung epithelial cells. It's a completely different organ.
Lung epithelial
Oh,
cells?
yeah. Sorry, lung epithelial cells, which is a completely different organ. But a lot of the literature is pointing to the importance of the vasculature and vascular development and even vascular progenitor cells for helping engraftment. I know it's kind of complicated in the eye, especially with like diabetic retinopathy, where you've got excess leaky vasculature. But I was wondering, what are your thoughts on incorporating vascular cells in the models of engraftment and migration? And
We're doing that right now. I didn't show it on here because I know that everyone wants to go on. But one of the projects we have now, so there's two barrier functions in the eye. There's the inner retinal blood barrier and outer retinal blood barrier. And so we developed a device that's looking at, particularly for microglia, looking at the inner retinal blood barrier and how we have vasculature on one side, microglia and retinal ganglion cells on the other side, and how we can get the transport across that to affect the neurons on the other side. The inner layer. So we've made a barrier. We've made a device. Of course, you made a device, right? With microfluidic glasses, we made a device to study that, and that's what we're doing right now in applying electric fields and seeing how we can use that to kind of repair the vasculature or hold it to at least stave off the leakage that causes vision loss.
That sounds really cool. Thank you.
Well, we got all the questions. Let's take Professor Vazquez again. Thank everybody for attending. We look forward to seeing you back here on January 21st.